Okay, so good evening, everybody. Heidi, we can jump directly to the next slide because this was just a presentation. So basically, today I will try to summarize very briefly what the experiments with the functional near-infrared spectroscopy is and what is the purpose of this study. First of all, the purpose. Basically, we want to identify an unbiased and quantitative outcome measure that can complement the clinical rating scales in the evaluation of the efficacy of new therapeutic interventions. So this is, of course, not the, well, we don't need just one quantitative outcome measure, but this can be one of many others. The first one, maybe that is completely quantitative and unbiased. Next, please. As you probably already know, this is a multi-site study involving 2 sites in Europe, the Stella Maris Foundation in Pisa, Italy, where I led a group of research, and we started already to do some fNIRS experiments, and a group in France, led by Dr. Aurore Curie at the Hospice Civil de Lyon, with the support of Extraordinaire Foundation, at least in France. And then there are 2 sites here, there in US, at Boston Children's Hospital, thanks to a collaboration with Michaela, and at the NIH, thanks to the collaboration with Dr. Samar Rahal and Danny Porter. And there, of course, thanks to the support of ACD Foundation. Next, please. So what is a functional near-infrared spectroscopy? It's basically a totally non-invasive technique that relies on the capability of the infrared light to penetrate the skin and the skull and allows us to visualize brain activity. In particular, we are measuring hemodynamic responses in the brain, which are an indirect measure of brain activity based on the consumption of oxygen that is higher in more active brain areas. As you can see from the picture on the left, basically there are many optodes, which are the sources of the infrared light and sensors, which are mounted on a textile cap, which is worn by the subject on the right. And then there is an additional shower cap just to cover everything from the environmental light. So this is quite, well, it's quite easy to wear and not so, it's quite comfortable to wear. Next one, please. What we are measuring are basically visually evoked responses. We devised this visual stimulating protocol. Maybe you can start the cartoon because there is a movie there. Okay, so
School. 5 more minutes.
This should increase the entertaining value of the visual simulation because there is a cartoon as a baseline. The audio is on, even if it's in grayscale, and the merging procedures allow us to still resolve the profiles of the characters and we can follow the storyline in any case, even when the checkerboard appears. The video is frozen. Oh no, it stopped, just stopped. Okay. So basically this is more entertaining with respect to the standard visual stimulus that is composed just by the gray baseline and the checkerboard. The cartoon should increase the attention towards the screen. Next one, please.
I want to go to school. 5 more minutes.
Yeah, and why we decide to do fNIRS and visually evoked fNIRS, basically relying on our previous results in the animal model that we obtained using intrinsic optical imaging, which is a totally comparable technique that we can use in mice. As you can see from this graph, very briefly, we have seen that the amplitude of these visually evoked responses is significantly different between animals with modeling CTD in blue and wild type littermates in gray. And in particular, the absolute amplitude is higher in the mutant animals. And this is true at different stages, at different ages, suggesting that this difference is stable across the disease progression, and this measure is quite reliable at the tester test. Next one, please. Okay, this is a brief video that I will let you watch without my comments below, because this video briefly summarizes the whole procedure. So the different steps of the procedures. You can start the video, Heidi.
Welcome to our lab. We are inviting you to come to our lab and watch cartoons together. It'll be a lot of fun. We will use a special cap to understand if you like the cartoons you are watching. First, it will take a couple of minutes to fit the cap properly. We will take a few minutes to adjust the cap. Please be patient and keep watching your favorite cartoon. You will wear a second cap to protect the first cap from extra light. Okay, now that we're ready, let's turn off the lights. One of your caregivers will be next to you the entire time. It is important that you always look at the screen and stay nice and still. Make sure you don't move. Sometimes you will see a funny checkerboard or hear a tone. Don't worry, just keep watching the screen and enjoy the cartoon. Each cartoon will last around 10 minutes, followed by a short break. During the entire session, you will watch 3 videos. If you prefer, you can choose any of your favorite movies, cartoons, or YouTube videos. Join us to watch your favorite cartoon. We'll be waiting for you.
Yeah, basically this video summarizes the more salient steps of the procedures. Here, of course, it's the setting in Michaela's lab at BCH that we will move to the where the will be done. Sorry, my connection is not so very stable. So next one, please.
Maybe remove your video, Laura.
Okay. Yeah, this is a very simple list of how you might prepare the participant before the day of testing. So basically, the idea is to frame the procedure as an enjoyable experience. For example, we usually tell them we are going to the cinema. We would like to see if you like our movies or something like that. And maybe some steps of previous familiarization with the movie in part, because there is the checkerboard inside the cartoon, or can be useful. I have a link. I have the link with Nemo that can be useful for that. And also maybe wearing a shower cap for some minutes, again, for familiarization with this kind of, having something on the head could be useful, but this is not mandatory. It's just some suggestions that can be useful. And for the day of testing, it's important to remind him or her that, well, today is the day for the cinema, or today is the day for the game. I don't know, referring to the experience that you mentioned when you talk to them about the experiment. The important thing is that the participants should wear the hair down, because we need to be able to adjust the cap in the proper way. During the experiment, only one parent or sibling can be present for the whole duration of the session, and can be sit close to the kid. And well, the last thing is very stupid, but a bathroom break before the session begins is important, because we have seen that sometimes the kids ask to go to the bathroom during the session. Next one, please. And this is the secondary objective of our study. Basically, in addition to see whether we can discriminate between CTD patients and neurotypical controls with the amplitude of fNIRS visually evoked responses, as we did in the animal model, we would like to test the hypothesis that there is a correlation between the amplitude of fNIRS signal and cognitive and behavioral deficits in individuals with CDS. And in particular, we would like to test the cognitive and adaptive behavior with the Vinal and adaptive behavioral scales. So we are looking to collect data from VABs and to make the correlation with the fNIRS signals. And again, we would like to do that because we have some important promising preliminary data, not preliminary, promising data from the animal model in which we have seen that there is a significant correlation between the absolute amplitude of iOS recordings and the behavioral performance in some behavioral testing, assessing cognitive function and motor function. And in this case, the higher is the amplitude of the iOS signal and the poorer is the behavioral performance. So this suggests that this biomarker can predict the severity of symptoms, which is an important thing for an outcome measure. Next one, but I guess I'm over with this presentation.
Thank you, Laura. So maybe now, the question is, how does this relate to the symposium? We had on the form, there's opportunities to participate in the study at the symposium or at Boston Children's, and you were able to indicate that it's kind of a 50-50 interest rate of those who signed up for today. But I just wanted to share from ACD about the symposium that there are going to be appointments available on Friday, Saturday, Sunday, and Monday. And you will speak with the research coordinator on Dr. Faggiolini's team to share your preferences with them on maybe you already have your flight booked and the only times you could do are the 28th and 29th, or maybe you would like to come into Salt Lake early. So you will be working out your participation with the research coordinator. ACD has arranged with Mercy Medical Angels to support half of the cost of the flight for one caregiver and one patient in a household to come participate in this. They will try to support international participation. They have to negotiate with their partners, but the process is secure an appointment with the study coordinator. So first of all, find out if you are going to participate and you're comfortable and you consent to the study. And when you're sure of that and you know when you need to be in Salt Lake, then look for your flight that you want to take and email that information to Kelsey. And Kelsey will work with Mercy Medicals to get the flight approved. They have stipulations that are very common, like you need to be flying coach class, not first class, and things like that. But you'll first get that submitted, we'll get it approved. And when it's approved, then you would book the flight and you would get a reimbursement for the 50% of the cost of your flights, the two flights, the caregiver and patient. And we realize that this is a little close to the symposium. And so if you've already booked your flight for the symposium, ACD will use ACD funding to cover one of your hotel nights at the symposium to help you in that way instead. We just want to try to eliminate some barriers and help you in some way so that this is doable for your family if you decide to participate. And if you did have a hotel night covered instead of the flight, you would be covering your hotel up front, and then you would get reimbursement from ACD by submitting a receipt. So now we're going to go through all the questions that everyone submitted in the forms, and then we'll check the Q&A box.
Sorry, one important thing that I forgot to mention is that during the experiment, the participant can watch the favorite movie or cartoon or video, basically anything they prefer to watch. There is no selected cartoon to be seen. We don't have to watch to Nemo, everybody, what we can choose freely the movie for the visual simulation.
So because of this, I think we would need to coordinate with you. If you are interested in participating, we need to know in advance which movie is the one movie, video, cartoon that you choose, because we have to download, integrate, and prepare. We cannot do at the same time when you arrive and do in that moment. So we do need some coordination in advance. So just think about, you can maybe make a list of 2 or 3 that you think are the best one, and then we pick the one that you think that your son or daughter really like to watch.
Okay. First question was, how many kiddos do you expect to be able to participate due to behavior attention issues? So maybe you have some experience, Dr. Baroncelli, from Italy and France. Like, what was kind of the success rate of these kiddos, and how challenging is it really to sit still and watch the movie?
Well, our previous experience is limited to the recording of 6 patients. For now, our success rate is 100% because all of them were able to stay still enough to have at least one recording completed. So we were very happy about that.
Okay. And will there be questions during the intake process about how attentive the child is, to kind of establish if you feel that they'll be able to participate, or are you pretty confident, like, we'll find a way?
Yeah, no, usually the idea is that we can find a way. We are pretty confident that the idea of using this this favorite movie or cartoon should be entertaining enough to induce the participants to look at the screen, at least for a limited amount of time.
Okay. Great, thank you. Is this study for both male and female patients?
Yes, in the long term. For the pilot part, we are just including males, but we are also planning to do females.
So most likely for the meeting in Utah will be specific for males, okay? For the second part here in Boston, we will expand also to females. We are doing in this way, not because we want to exclude anyone, it's just because it's a pilot, we would like to achieve significance or data that are more consistent. So maintaining the same sex, same gender is helps in that direction. So we don't want, if someone is interested and is the parent of a female, please keep us in mind, and hopefully we can have you come here to Boston Children's Hospital.
Would you say that first of all, would you say that this stipulation only applies to CTD because of the X-linked inheritance of CTD? Are you only accepting males with GAMT?
For now, yes, just to reduce the variability of the pilot study, but just for now, it's not something that we want to maintain in the future.
And how about if someone's joined us and they do have a daughter with CTD? Should they go ahead and reach out to the study coordinator to express their interest for the off chance of, if we don't have enough males to participate, they can let you know of their willingness to participate? Or is it a pretty firm no females?
So right now our IRB lists males as the subject, and as you know, we are in the process of getting the approval. And so once we have the approval, we can amend it to include the females, because we wanted to secure the approval and make it as smooth as possible, as faster. So right now, so I would say, if you're interested, if you're parents of a female subject, please keep us in mind. Once we have all the brochures and all the approval, we will reach to you as well. Okay, so you can express an interest and specify that you are the parents of the females, and then we will reach out to you once we have the approval for coming.
Okay, thank you. Will I get a copy of results and an interpretation of what those results mean?
Well, the copy of the results, there are no well, the main result that we have is the latency and the amplitude of the response, the visually evoked response. So of course we can share how the experiment has went, basically, but it's not so meaningful to make an interpretation of single results, at least at this point, because we need to understand whether there is a difference between patients and controls. And only when we have this result, we can see whether there is a correlation between the amplitude and the severity of symptoms. So we can, of course, discuss about the experiments, but I'm not sure that there is an actual copy of results that can be shared. Of course, something about the interpretation, yes.
Yes, but usually for these studies, we is one individual subject that will, it's difficult to give an interpretation just from one, as you need to have collected everybody and then publish the work. So it's not for, I will have my recording and that will be immediately translated. So this is for the bigger picture of the establishing the biomarker.
Okay, thank you. Will I get to do this again one day and see the change after a new treatment? So I think, again, following along with, learning about how this is relevant, and it might be a little early, but do you plan to repeat with patients? And
yes, the idea is to assess also the test-retest reliability, and of course, would be great if we will find a difference between patients and control to see whether a treatment can modulate the biomarker, because this is just the purpose of the study. Again, in the animal model, we are seeing that this is the case, that the biomarker can be modulated, but the amplitude of the biomarker can be modulated by a treatment with at least partial therapeutic efficacy. So the answer is yes, basically.
Yeah, I would just wanted to add, we are at the stage we are establishing this, we are trying to establish this as a functional biomarker for CDD. And so the first step is to run experiment, collect enough subject, neurotypical versus CDD subject, see if we confirm the difference that we have seen in the animal models. That means that it has to be published, and the FDA has to sort of accept this as an outcome measure, a readout. So there are some steps, but the goal is, this is the first step, and then hopefully we can use this in a clinical trial of a gene therapy, an ASO therapy, a protein therapy. That's not what we are doing here, testing a therapy. We are establishing the biomarker and see if this biomarker will respond. Okay, so ideally, people who will participate in this study will have a baseline, and if they participate in the clinical trials, and the clinical trials add the NIRS as an outcome measure, they could see before and after. But I can tell you, if this get approved as a biomarker, functional biomarker, and included in clinical trials, you will do again a baseline before the treatment and after.
Thank you. Do you have recommendations for selecting videos?
No particular recommendations. The only thing is that the video should be something that your son or daughter likes to watch.
Okay. How still will my child need to be, and for how long?
Well, one recording section lasts 10 minutes. We have some pipelines for movement, artifact detection and correction. So of course, ideally, the participant should be still and should watch to the screen at least when the checkerboard is coming. But some movements of the head can be tolerated. Basically, they cannot get up from the chair and start moving around the room because they wear the cap, but the cap is still connected with wires to the amplifier and the recording system. So this is not allowed, but some movements of the head are tolerable and can be corrected in the post-processing phase.
Maybe what we don't want is that they use their hands and touch the net. That of all the movement, the little movement, we can also exclude from the recording post after the recording itself. But if they start to touch the net, they will move the electrodes, and that will compromise the recording. So in that sense, it has to be a little more quiet.
Mm-hmm.
Okay. So some questions from the Q&A box. Is this study only for children or adults as well?
Yeah,
The study is open. It's probably we will need to analyze separately young.
Age.
Yeah.
But for now, we want to record everybody.
Everybody.
The more we can record it, the better it is.
Mm-hmm.
Okay. When will Boston/NIH begin scheduling? How does hotel/flight work with doing it separate from the symposium?
So this is a great question, and we hope to start as soon as possible. I would say that definitely not before the symposium, so it will be after. And regarding NIH is already recording actually patients so this is in Bethesda. Maybe we can put you in contact with them if that is easier for you to reach their location than us. Regarding hotel and flights, we had applied for a grant asking specifically for support for the families for traveling and lodging. We don't know the answer yet, but we are trying very hard to secure funding for everybody, so it does not become an expense for you. At this moment, we don't have the money, but we will find the money.
So it sounds like if they reach out to the study coordinator after this meeting, it's not a firm, commitment and schedule, and you're expressing interest and they're just getting in touch with you, and then you'll let them know how everything shakes out with supporting travel and hotels and things like that, right?
Yeah. Because our IRB is still under review, we cannot reach out to you. You can reach out to us or reach out to the ACD. Okay, and we are very happy to have a database that we can then reach out. But right now, it's not legal for us to reach out to you.
Okay. We covered this, no age limitations. And then last question. This patient is on the more severely impacted side of CTD. They're nonverbal, limited attention, wiggly, does not always like something on their head. Would it be appropriate for them to participate in this study?
So we already did a recording at least one recording or maybe two in nonverbal children. The span of attention, well, let's say that it depends what limited means. Of course, if it's less than 1 minute can be an issue. If it's, let's say, at least 5 minutes could be good, because we can also shorten a little bit the recording and average less trials. About the problem with something on the head, again, it depends, because as Michela mentioned before, they should wear a cap and they should be able to avoid touching the cap and the optodes. So this is quite
I would
important.
say maybe if you're interested, I would practice wearing a shower cap, maybe a little soft hat, and then a shower cap on top while watching videos. Okay, so do at home already something like that without saying anything, okay, but that it could familiarize and relax a little bit, that's not so unusual. I think it's going to be much more challenging if you never do that, and then you arrive and then you ask in this new environment with new people and say, oh, put the cap, stay, don't move, do this, do that. Any child will not comply to that. So some preparation, it will be strongly encouraged. And that's again why we are saying about choose the best video, that not that we like, that the subject likes, not that you like it. It could be the one that they're watching over and over and over and drive you crazy, but they will always be capture with their attention. So I will suggest, if you are interested in participating, do these little things in advance. That can help a lot.
That's great. One parent shared that they picked their child's favorite thing, which was playing the iPad. And for a while to prepare, they were saying, "Oh, put the cap on, and now here's your iPad." And so it was that continuous positive reinforcement that it was fun to put the cap on, because then you got to do your favorite thing, and then it worked really well. I saw. Oh, we do have one more question now. Let's see. Would the parent be allowed to help still the child's hands?
Yes, one parent can be sit close to the participant and be there. The ideal thing is that they stay silent, just to avoid that the kid moves the head towards them, but they can, touch and comfort them.
Yeah.
Without
so
any problem.
it's part of the idea of being comfortable while you're doing the recording without disrupting the recording. The focus should be on the watching the video, and you are there to calm or to keep calm and comfortable your child.
Yeah.
Okay. And you mentioned giving the families a link to the video. Are you wanting them to watch before the testing? You're wanting the patient to watch the video with the checkerboard?
Yes, just to avoid any, let's say, surprise effect during the recording. Could be a good idea. At that link, there is, I think, 3 minutes of Nemo with the checkerboard randomly appearing during the cartoon. So could be a good idea to familiarize with this kind of cartoon, which is a little bit unusual because you have the audio on, but it's in grayscale, which is, again, something that can surprise a bit, in particular younger ones. And the checkerboard can be also, well, probably not a wow effect. So maybe some familiarization in advance could be useful.
Okay. Well, we have
I can share with you, of course, and I can share the link with you, and you can share with those are interested in.
Would you be sharing then just the Nemo link, or are you, if like someone says my child's favorite video is Frozen, are you preparing Frozen to be similar and sharing the Frozen link, or is just
I can also do that, of course. or yes, I can also do that. Or I have already prepared the link with Nemo just to familiarize with the checkerboard. But of course, if Nemo is completely wrong choice, I can add some more examples.
Yeah.
Okay.
So what we are saying, this is just an example to give an idea of what is coming. We do need to know the video of your child. So the sooner we can have, even if you're not coming right away, Laura will prepare all of that. And then, of course, if they're ready, we can share again with you. But the goal is to have the good one for the day of the testing. And this Nemo was more to, so you understand how really it's 3 minutes and how it looks like that, more than, oh, let's watch Nemo.
Mm-mm.
Okay. Okay, well, we have up on the screen an email address that the families can email to express their interest. And based on that, we'll work with everybody on slots at the symposium, and then also airline or hotel supports. And then we'll look to stay in touch on what supports might be available for participating at Boston Children's. And just you can email this address for any questions. Doesn't have to be that you are ready to do this or sure and certain that you're doing it. The lines of communication are open there. You can reach out to us at ACD and we'll help get you the answers that you need. And
So if you write to Adeline, maybe you want to say the age of your child or adult son, daughters. And if you have already some preference or some no, please let us know, because that already it can help us to create a database for us to work with. Thank you. You don't have to share private information. Okay, so I don't want private information. It's just a really a very basic to facilitate our organization for the experiment.
Yeah, and if you have any additional questions in the next weeks, you can share my email without any problem, and I will answer. And I guess the same for Michele.
Yeah. So we are here to work with you. Okay, not you're helping us. So thank you.
Yeah.
okay. It's very exciting. Thank you so much for your time and for everyone that joined, and we'll share this video online if you want to refer back to it so you can share it with the other caregivers and let us know how we can help. Thank you.